Okay guys, today I'm going to be breaking down some AP biology questions. As you know, this year, 2020, the AP exam is online. It's 45 minutes and it consists of just 2 questions. One of them is a long free response question and one of them is a short free response question. But in this video, I am just going to break down a short free response question. Okay, so any free response question that you have, I usually complete them in 4 steps, okay? The first step is to identify all of the questions. Okay, and what that means is most AP questions have multiple parts. So there's a part A, there's a part B, there's a part C. So I want to find all of these and recognize how many parts. Okay, that's always my first step before I even start writing things. The second thing that I do is to identify all of the command terms. Okay, and command terms are things like justify. There are things like predict, describe, connect. These are the command terms. It's telling you what to do. Okay? So it tells you what to do. It tells you how to answer the question. Okay, after I have identified all of the parts of the question and after I've figured out what the command terms are, that's when I go back. Step number 3 is I read everything. Read everything. Okay, you got to read everything with the questions in mind. Okay, so you're going to read everything. Read everything with these 2 steps in mind. The last and final thing that you do, this is step number 4, is to just use evidence and data if it gives you to respond. Okay? And you're responding to each question. Okay, so now what I'm going to do is look at this question. This is an example of a short free response question. And I'm going to follow my steps. First thing I want to do is to identify all of the questions. So I'm going to skip this for right now and just look here, A and B. So there's 2 questions. Okay, question part 1 is using the model of the P53 MDM2 pathway. Connect the reduction of MDM2 function in mutant mice to the observed concentration of red blood cells. Okay, so I'm going to be looking at this model and I'm going to find something. I'm looking for something about red blood cells. Okay, part B says to predict how a loss of function mutation in the P53 gene is most likely to affect the cell cycle in response to DNA damage, and justify your predictions. Okay, so now I'm moving on to step 2. Step 2 is to identify all of the command terms. In part A, there's 1 command term. I'm connecting the reduction of MDM2 function in mutant mice to the observed concentration of red blood cells. They're going to tell me something about red blood cells. Okay, part B, the command terms are first I need to predict how a loss of function in the P53 gene is most likely to affect the cell cycle in response to DNA damage. Then the second command term is to justify my predictions. So I know that in part A, there's 1 thing that I need to do. In part B, I have to do 2 things. The AP biology tests make it really clear with what the command terms are. They're in bold. That's what the question is asking you. So I also already know that this question is worth 3 points. Okay, it's worth 3 points because there's 3 things that it's asking me to do. All right, on to step number 3. In the description below is a link to find the PDF version of this question. So I'm not going to read the whole thing, but I'll just read some key things. So pause the video, read the whole question with the command terms in mind. Okay, so now that we've read through the question with the command terms in mind, we are ready to move on to step 4 of this question. Okay, so for part A, I want to connect the reduction of MDM2 function in mutant mice to the observed concentration of red blood cells. Well, let's look back and see what MDM2 is doing. The P53 and MDM2 proteins are involved in the DNA damage response pathway. Okay, and it's okay, I'm going to annotate that, and that's this figure. In the absence of DNA damage, MDM2 mediates the degradation. Okay, what's this word? Let's read on and see if it explains. The degradation of P53. In the presence of DNA damage, P53 is not broken down. Okay, so DNA damage, P53 is not broken down and forms active tetramers that stimulate, okay, it stimulates the expression of genes that block the cell cycle progression. Okay, so what I understand about this model is that when there's DNA damage, P53 is not broken down, and it stimulates the expression of genes that block the cell cycle progression. Okay, so using the model, let's see if we can connect. We have to find now about red blood cells. So this is background information I learned, and this is the application. Red blood cells are produced by the division of differentiation of multipotent stem cells. Researchers have observed that mice with partial loss of function mutation in the MDM2 gene have a lower concentration of red blood cells in the blood than do mice that express the wild type MDM2 allele. Okay, now we can start to link some things together. So, knowing that MDM2, when there is no DNA damage, causes P53 to degrade or to break down. These mice have a partial loss of function mutation in this gene. So if this gene is causing P53 to break down, what's going to happen is that less of these are going to be broken down, and if they're not broken down, they're going to activate these tetramers, and if these tetramers are active, they're going to block the cell cycle. So that's what this is saying here. For part A, one thing that you could say is that P53 is always active in the MDM2 mutant. Okay, we could make that connection. And I'm connecting that because, well, if P53 is active, it's going to stop cell cycle production and probably stop producing red blood cells so that we have this lower concentration of observed blood cells. Okay, another thing that you could do, and you just need one, is that you could say, in other words, P53 is not degraded. Okay, it's not degraded regardless of DNA damage. Okay, so either one of these things, you could say that P53 is always active, which is causing the cell cycle to be blocked, or you could say that P53 is not degraded, even though there's no DNA damage. Okay, now it's time to start answering part B. Part B. Okay, So we want to predict how a loss of function mutation in the p53 gene is most likely to affect the cell cycle in response to DNA damage. Okay, so now here is where we need to have two things. We want to have a prediction. So let's think. There's a loss of function mutation in the p53 gene this time. So the p53 gene, what does it do? It blocks the cell cycle production, and ideally when there's DNA damage. So in this instance, how might a loss of function mutation, so p53 doesn't work, how is it most likely to affect the cell cycle in response to DNA damage? So let's make a prediction. Okay, so if it has a loss of function, perhaps it won't do its job when there's DNA damage. Okay, that could be a problem. The justification now of this would be explaining why this is the case. So we would say ultimately that the cell cycle is going to proceed if there's DNA damage, and the justification is that non-functional p53 protein, what it can't do is it cannot regulate the cell cycle. Okay? So we broke this down in 4 ways. Step 1 was to first identify all of the questions. Step 2 was to look at what the command terms are. After we identified what it is we're looking for, we went back and we read everything. We looked at the diagram, we read all of the text. After we've done that, with these things in mind, we were prepared to answer the question completely with detail and also to be concise. So hopefully that helps as you break down these complicated free response questions and to understand that it's really just doing things in pieces. Yeah, so hopefully that helps. Anyways, check out the other video that will be linked on breaking down a long free response question. That's it. See you later.